Okay, I have a kind of travel thing to do, so we have to switch to virtual for this class. I'll hit you up on Blackboard about Friday. I'll probably be out on Friday as well, and I think this video will take us through Friday's lecture, but I would like to meet synchronously then, if possible, to kind of set things up for the next chapter. Um, but we'll just play it by ear. So just be on the lookout for Friday's lecture. This will be for Wednesday, January 22nd's lecture. So last time we were talking about the kind of things that you already know that are kind of required to understand biochemistry. We talked about the types of chemical bonds and how they all interact, what the nature of those bonds are. And I then asked this question about whether two complementary strands of DNA will spontaneously bond together, and how do those chemical bonds contribute to that process. So that's something we should be thinking through and should be able to answer. The other thing to kind of think of in terms of biochemistry is that this is the chemistry of life. Here on this world, in this universe, life is carbon-based. That might not always be the case, but for this particular one, it is. And then, so carbon forms the backbone of all biological um, macromolecules um, and organic chemistry. And then there are other elements that are predominant there: hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the most common. But then there's so-called vitamins, small molecules, metal ions, potassium, sodium that are all critical for life, but are found at much lower concentrations than these big ones. So about 30 elements out of all of them are necessary for life. Here are the big players over here. Oh, I can paint. So big players over here and here, and then the vitamins over here. And some point we'll talk about nutrition, and I'll talk a little bit about how we discovered vitamins because it's a bit interesting. So we do a little bit of organic chemistry. We're going to go kind of quickly through this. But the most common bonds that we're going to see are carbon bonds: the carbon-carbon bond, the carbon double bond, the carbonyl. Um, that's important. And then some of these other types of carbon-based bonds. We know from our general chemistry that covalent bonds are shared pair of electrons that are denoted by that single line. So obviously, the carbon-carbon double bond would be four shared electrons or two pairs of electrons out of there. And so here are some of the common ones: carbon dioxide, C double O, carbonic acid, so forth and so on. And we know there's a little stereochemistry that comes into play here for carbon-carbon single bond. There's free rotation around that carbon bond. And so these can kind of spin, like a wheel on an axle here, and that you form this tetrahedral shape because there are four valence electrons in carbon that can all contribute to pairing. The reason that carbon is the backbone of organic chemistry is because of this four valence electrons and the shape. This allows for two pairs of electrons to propagate a chain of carbons, so you can continue to make macromolecules, but also gives you two other electrons that can either branch off chains or add different flavors of chemistry. And we know that in the carbon-carbon double bond, where you share two pairs of electrons, that you also kind of fix a planar shape there. And because you're sharing these two pairs of bonds here, you don't have the freedom of rotation that you do in a carbon single bond. So if you have a carbon-carbon double bond and four different functional groups attached to that double bond, you actually have a fixed planar shape there, in which by breaking this double bond and reconfiguring one of these functional groups, you have two different flavors of the same compound. So these are isomers of each other, right? Um, Same formula, but yet because of the orientation of these four different functional groups, then you have two different outcomes, a cis and a trans outcome. Y'all should remember that from organic chemistry. And if we look for this carbon-carbon single bond, even though we say that there is 360 degrees freedom of rotation around that single bond, that's actually not the case. There are some energetic maximas and minimums that are in there. And if you note, an energetic maximum here is when these electrons line up again together. And then there is an energetically favored position denoted by the lower delta G here when these hydrogen atoms are actually aligned in such a way that they have the most space between them. And so even within this kind of quote unquote free 353 range of freedom on this carbon-carbon single bond, there actually is some favorable positions here that maximize the distance between the hydrogens in this case on this bond. And so the cell can actually utilize these differences in order to form an energetically favored three-dimensional shape. So I mentioned before that one of the kind of most important things that we're going to take from organic chemistry, besides mechanisms, are going to be the use of functional groups. All of these functional groups that you see here, y'all should be familiar with from organic chemistry. And we'll use some of those chemical properties quite extensively, and we'll see kind of some of these over and over again. There are things like the carbonyl. This is an aldehyde in which you have a C double bond O at the end of a carbon chain, right? That is like glyceraldehyde 3-phosphate. So a lot of our sugars kind of end in this carbonyl. Also have a ketone in which you have this in this aldehyde. Ketone in which you have this carbonyl that has a carbon chain on either side of it. So the carbonyl is buried within the structure. So we'll see that quite a bit. Let's see. Phenyl group, we'll see particularly in the amino acids like phenylalanine and tyrosine. An ester will form, we'll see some of these esters, a thioester in particular. We'll look at acetyl-CoA, which is one of the kind of most prominent biomolecules in metabolism. The amino group obviously is going to play an important role in amino acids. We'll look at this enol group, and that'll actually be important for a lot of the mechanisms that will cause the movement of these electrons and change in structures. The imidazole group is an important part of the amino acid histidine, which in and of itself is one of the most biologically important amino acids that are out there. And so forth and so on. Disulfide bridges, thioesters. So we'll see all of these, and you should have some sense of the basic chemical properties of these functional groups as we engage in the material. And what we do with biological macromolecules is really just kind of string together a bunch of these different functional groups to have, a super ordered function. And so here, this is acetyl-CoA. Acetyl-CoA is actually, acetylated coenzyme A, and it is, one of the kind of key convergent points on catabolic metabolism. We see this at the top of the citric acid cycle. And in fact, the whole purpose of glycolysis is ultimately to produce these acetyl-CoA um, molecules. And you can see that these functional groups that we talked about before form this. And one of the things that we should kind of get used to doing is just being able to identify the base macromolecule from, a structure. And so there will be points on the exam where I just give you a lot of structures and have you identify it. And you should be able to, even if you kind of aren't familiar immediately with what the structure is, at least identify what its foundational biological macromolecule is and um, and then kind of what the modifications might do. So for instance, looking at this acetyl-CoA, right, and we know we have four biological macromolecules: the lipids, nucleic acids, amino acids, and carbohydrates. I could ask you, okay, so what is the base biological macromolecule of this? And you should be able to identify relatively quickly that this has a ribose sugar, it has a nitrogen containing base, and it has phosphate groups, so that this is at its base a nucleic acid. And actually the A in coenzyme A is simply for adenine. And then attached to it, instead of that third phosphate group, there's this chain of other things, hydroxyl, this amino group, another amino group that ends in this thioester. And this thioester actually is the key to acetyl-CoA because it takes these acetyl groups and presents them to the citric acid chain to break down and pull out the energy for it. But, looking at this whole thing, even if I just showed you the structure, if you didn't recognize this as acetyl-CoA, I would expect you to recognize that at least it is a modified nucleic acid in this particular case. You can do the same thing for carbohydrates, lipids, so forth and so on. Um, and then here are the biological macromolecules that we talked about, the lipids, which are characterized by long chains of hydrocarbons that tend to be hydrophobic. Um, the carbohydrates, which are hydrates of carbon, and so they are characterized by having a 1 to 2 to 1 carbon to hydrogen to oxygen ratio. So CX H2X OX. Nucleic acids, again, nitrogen-containing base, ribose sugar, phosphate group, and then amino acids, which have an alpha carbon, a central carbon, a carboxylic acid group, a amino group, a hydrogen, and then an R group, in which can change the chemistry. So those are the base biological macromolecules. And again, you should be able to identify kind of common features of those from any type of modified macromolecule. So we talked again about that those carbon-carbon double bonds and that they have stereochemistry that's associated with them depending on the orientation of the functional groups that are attached to the carbons. And so for across the carbon-carbon double bond, you can have two different configurations, a cis and trans configuration. And those configurations, even though their formula is the same, can have dramatically different chemical and biological um, functions. And so here's an example. This is related, but you have retinol here. Retinol, as you can imagine, sits in your eyeball, right? And it is one of the compounds that are responsible for transmitting, for passing on the light information from, obviously the energy of the light. And so it sits in this cis conformation normally. You can see that double bond right here. there's a little bit of strain on that double bond. And so the energy of light is actually able to break this bond and switch the cis configuration to the trans configuration. So you can see carbon 11 and 12 here. You've gone from cis to trans. You flipped the position of this functional group. You straighten out this chain, and that sends a signal to the cell that light has hit that particular region, and then your cell interprets that as ultimately information that can be coded as vision. All right. So obviously, without those two different configurations, the body wouldn't be able to recognize the light that's processing at the back of the eyeballs in this case. And there are other kind of uses or utilities as well. This is the so-called trans fats. This was something, when I grew up in the 80s, a lifetime ago, trans fats were great. I was raised only on margarine and Crisco, which is basically just trans fatty acids. And so the reason that we liked trans fats at that time is because you could take plant oils. Plant oils are usually unsaturated fatty acids, and unsaturated means saturated with hydrogens, and so there are double bonds in these plant oils. So this is oleic acid here, and you see this double bond. That double bond causes a kink in the fatty acid, and that actually makes it liquid. That's the difference between oil and many fats. It's liquid at room temperature, and it also makes it much less stable. So these, will go bad if you leave olive oil out or something like sesame oil. If you leave that out, it'll go rancid relatively quickly. Saturated fatty acids do not, because there's really no place for chemistry to occur. And so saturated fatty acids are really very shelf stable. The most common one at the time, pre-trans fatty acids, were things like lard, based from animal fats, very expensive to make. Obviously, you need animals in order to make it. And so nutritional scientists got the bright idea that if you can convert this cis double bond to a trans double bond, it is a lot straighter of a structure. This is a lactic, acid here. And this actually looks a lot closer to a saturated fatty acid like butter or lard. It turns out that this is much more shelf stable, cheaper to make. And so we thought at the time that this could be kind of a really, easy transition to make, minimizing food cost, increasing the stability of it without much downside. We learned later, though, that in nature, we have evolved a system by which to handle a cis double bond in a fatty acid. And when we break down fatty acids by beta oxidation of fatty acid, there's a whole process by which we handle the double bonds that occur from mono unsaturated or polyunsaturated fatty acids. We haven't evolved a system to deal with trans fatty acids. And so when you have a trans fatty acid, what happens is that the cell processes these fats and they basically clip off two carbons at a time to form acetyl-CoA groups or acetyl groups. And so it does the beta oxidation of fatty acids two carbons at a time until it hits this trans bond. And then it gets stuck on the trans bond and can't do anything with it. And so it's hypothesized that what happens is that the body leaves these fragments of fats, floating around in the bloodstream where eventually they accumulate on blood vessels and contribute to heart disease and arteriosclerosis. So, mixed blessing, kind of the danger of knowing just enough science to be dangerous, right? Which, truthfully is the story behind nutritional science in general. but that is a subject for when I'm not being recorded. Let's say that. Okay, so another kind of key thing that we need to understand or at least appreciate for biochemistry is, the concept of stereoisomers. And again, we learned in organic chemistry about chiral carbons, right? A chiral carbon is a carbon that has four different functional groups attached to it. And when that happens, you can actually have two stereoisomers of that chiral carbon. The stereoisomers are the same chemical formula, but they have two different shapes. And so in this case, in alanine, this is the alpha carbon for this amino acid, carboxylic acid group, nitrogen amino group, your R group, which in this case is the methyl group, and hydrogen. And you can have two different flavors of this. Remember, stereoisomers, you kind of are like your hands. They are mirror images of each other, but they are not superimposable, right? So if you take your hands, they mirror each other, but when you try and stack them on top of each other, obviously the thumb doesn't line up with the other thumb. So uh, for our amino acids, 19 of the 20 of them have at least one chiral center, one chiral carbon, the alpha carbon. And so because of that, you can have two different flavors of those amino acids, L configuration and the D configuration. And we'll learn when we look in amino acid structure in more detail, kind of how to differentiate between those two. And certainly you will be tested on that. For our purposes, though, it's important to kind of consider that in nature, you really can't have a mix of L and D amino acids floating around biologically because you would not be able to form any consistent shapes and structures with two different orientations of amino acids. And so life had to choose one configuration, either the L or the D configuration, and just go with it. And in this planet, in this universe, life has chosen the L configuration, right? So at least for our amino acids, for most of our biological macromolecules, you're going to see the L configuration for those biological structures with some rare exceptions. But it could just well be, there might be a parallel universe in which we are the same, except our proteins are all in D configuration. There's no reason why that is not the case, although some scientists have speculated about, little thermostability, type issues. But there could be a planet in which life abounds in the D configuration and not the L configuration. And there actually is some biological relevance with that. Maybe when we're in person, I'll talk about a drug that kind of ignored their stereoisomer kind of chemistry and caused some profound developmental issues because of it. But we'll discuss that later. In general, compounds in that deconfiguration are not biologically active. And the reason is because in general, the receptors that are recognizing the, biological macromolecules are also in the L configuration. So in some sense, that makes sense. There are some instances. So here, if you look at this compound in the R configuration, or L configuration, this would be carvone, which is spearmint, so it gives you that minty flavor. And then in the S configuration, it gives a flavor that's like caraway. And I don't know, I might bring some caraway seeds, but this is something, if you ever had a Chicago hot dog, a lot of times they're topped with these caraway seeds. It's kind of like a fennel type flavor. So it's definitely not spearmint, right? You can definitely tell. So same chemical compound, right? The only difference is the configuration of that chiral carbon here, but the receptors on your taste buds actually recognize the difference between those two configurations and differentiate from them. Okay, I'm going to pause. I'm going to stop this one. This will kind of be the end of Wednesday's lecture. I'm going to go ahead and tape the thermodynamic piece, which will basically be Friday's lecture, and I'll post that up there, and then we'll kind of figure out whether it will be synchronous or asynchronous on Friday. Okay, so I'm stopping this now, and then